And I should add also, Art Levin was originally going to give this talk. He was unable to attend, so I'm speaking in his place. He did very kindly provide me with a number of slides, and I edited them freely. So if there's anything you don't like there, it's probably one of my edits. I have a very ambitious agenda for the next half hour. What I'd like to do is to try to give you an overview of the safety issues in oligo development, oligo ADME, oligo PKPD, and applications in clinical development. Obviously, I'm going to be summarizing a lot of these points. I'm going to spend most of my time, however, talking about safety issues in oligo development. And throughout my talk, I'm going to try to focus on issues that are unique to oligonucleotides, single-stranded, double-stranded, or other. The first type of effect from safety is due to Watson-Crick hybridization, and on-target effects due to the target that you're intending to hit, so-called exaggerated pharmacology. In addition to that, you can have off-target effects to hybridization of partially complementary targets. These are the unintended effects. And those tend to be worse with gapmer oligonucleotide designs than with mixmers. I think what may be happening there is that when a gapmer hybridizes to something that is partially complementary, it can activate RNase H and cleave it, creating an irreversible effect. Whereas when a mixmer hybridizes to a partially complementary target, there is no permanent effect. Regardless, empirically, just testing different oligos, mixmers tend to be much less toxic. These toxicities may not be detected in animal studies if the off-target gene is not conserved between man, rodent, and primate. During screening for sequences, I'll come back to this, you prefer sequences that are conserved because otherwise you may wind up in the situation where you have to design a surrogate oligonucleotide for your safety studies to hit the target in your tox species. These kinds of toxicities are most likely to be seen in the liver and kidney. Those are the major tox organs in animals, and it's probably because of the very high oligonucleotide concentration there, as Brett alluded to earlier. And for that reason, I think there's a possibility, at least, of detecting those if you do some of your preclinical screening using liver and kidney cell lines from humans. This is not always done. Now, it's rare in human clinical trials to have a toxicity show up that was not seen in animal studies, but it has happened, and on at least one occasion that I know of, it led to a program being terminated in phase 1 human clinical development because of a toxicity that had not been predicted in the animals. And so I think it's probably not a bad idea to do these safety studies in human cell lines. And now a quick thought experiment for you. And you don't have to answer this, but for a given chemistry, what is going to be more specific for the target? A longer oligo or a shorter oligo? Now, most people who are used to using oligos as probes for gels and blots and so forth say, well, a longer oligo is going to be more specific. But in fact, the shorter oligo is the one that is more specific for a particular chemistry. If you have a chemistry that requires 12 bases of complementarity to have high enough hybridization to hit the target, then a 12-mer is going to be only binding to that one perfectly complementary region, whereas a 20-mer could have multiple different areas that it could bind to. So I think there's a reason to try to design shorter oligos. There's also, of course, a reason to use newer chemistries that allow you to do that with higher affinities, but there is a bit of a trade-off there, and I won't get into all of the bioinformatics and so forth around that. Next issue I'd like to spend a little bit more time talking on is the effects due to protein binding and immune stimulation. So these are not the Watson-Crick hybridization effects of the oligos. And the issue there, as you've heard a little bit about already, is that phosphorothioate oligos are very highly protein bound. In vivo, more than 90% protein bound to serum proteins. And a variety of these protein binding interactions can lead to unintended and usually undesirable immune stimulatory effects. The CpG motifs in DNA are particularly well known. That's mediated through a receptor, toll-like receptor 9. Certain single-stranded RNA sequences can activate toll-like receptors 7 or 8. TLR3 can be activated by double-stranded RNA, but most studies say that you need at least about 50 or 60 bases of a double-stranded region to activate TLR3. So it's probably not an issue for most of the synthetic oligonucleotides. For mRNA, it could potentially be. Poly-G regions, if you have 4 Gs in a row, or even just 4 out of 5 bases consecutively G, these can form so-called G tetrads, which can stack into G quadruplexes. They can have a lot of different effects. Most people designing oligonucleotides just use algorithms to screen out any G-rich regions to avoid that. 2' modifications at the 2' fluoro modifications can increase protein binding, but really any negatively charged oligo, especially phosphorothioate, you do have to expect a high degree of protein interaction. Now, TLR activation is not the only type of immune receptor that can be activated by nucleic acids. In fact, there are quite a variety of other innate immune receptors that can be activated, but the TLRs are the best studied and the best understood at this point. The TLR activation is not detectable with neutral backbones, which is nice. It is reduced by CpG methylation, but it is not completely eliminated by CpG methylation, and I'll show you some data on that. The other thing that I'll just mention is that it's not just the CpG itself, but also the bases around the CpG that determine the stimulation. And in the case of RNA immune stimulatory sequences, those can be greatly reduced, if not eliminated, by various 2' modifications, the O-methyl, the methoxyethyl, and so forth. And in the case of mRNA, of course, there's been a lot of work done on modifications to reduce mRNA immune stimulatory effects, a lot of literature on that I won't have time to go through. The toll-like receptors that detect nucleic acids are located in the endosomes of cells down here, and they have evolved to detect pathogens based on pathogens coming into cells and the nucleic acids of pathogens being uncovered in the endosomal compartment. Normal nucleic acids are excluded from those compartments. So the immune system has put receptors there that if these receptors are activated by nucleic acids, it's most likely a pathogen. And that triggers a variety of immune stimulatory effects through pathways that have been reasonably well identified. In addition to these effects of oligonucleotides, there are a variety of other proteins that have been reported to bind oligonucleotides with high affinity, and quite a variety of biologic effects that have been characterized to differing degrees. These effects are typically concentration dependent, and so again, going back to the point that Brett made, using newer chemistries that allow you to reduce the concentration of your therapeutic oligonucleotide, you're less likely to encounter these effects, but it certainly behooves the drug developer to be aware of these effects if you're working with that type of chemistry. This is just to show you that CpG methylation does not eliminate immune stimulation. You see on the left-hand side a controlled CpG oligo. This is just measuring B-cell stimulation as a simple assay proliferation. If you methylate the CpG, you reduce but do not eliminate it. A GPC further reduces the stimulation, but this is just background stimulation by any phosphorothioate. At high enough concentrations, they will trigger proliferation of B cells regardless of the sequence almost regardless of the sequence of the phosphorothioate. Likewise, other sequences can be stimulatory, CpG-free sequences. What you're looking at here are 3 different oligonucleotides. The first one on the left is just poly-T. The next one has a single C inserted at the second position, and then you have a single G. The point of this is that polypyrimidines turn out, especially if you have a few C's sprinkled in, to have immune stimulatory effects that are mediated through TLR9 in addition to the usual effects. And those depend on what assay you use to detect stimulation. So this is IL-10 production. IL-10 induction is a relatively promiscuous effect of phosphorothioates, that many different sequences will stimulate that. Whereas interferon alpha production is much more rigorously controlled. You have to have a CpG motif to induce that. These are different cell types that are responding. The interferon alpha is coming from a kind of dendritic cell called plasmacytoid dendritic cell. The IL-10 is coming predominantly from B cells, but also from monocytes and macrophages. Don't want to go too much into the immunology, but with RNA, likewise, different sequences have very different stimulatory effects. This manuscript was one of the first ones to show that, was one of the first ones to show that just using beta-gal as a target and making a variety of different RNAis against beta-gal, and these are unmodified RNAis. Some of the sequences induce very high levels of interferon alpha production, others induce almost none at all. Further studies have shown that there are a variety of different RNA sequence motifs that selectively activate either TLR7 or TLR8 or both. And this is just showing some of the sequences that have been described there. TLR7 is expressed in plasmacytoid dendritic cells and B cells. TLR8 is expressed in a variety of other cells. And so you get different cytokine profiles, interferon alpha or TNF alpha or IL-12, depending on which of these TLRs are activated by your particular RNA sequence. Now, if you just randomly make an RNAi against anything, on average, about 80% of a 20-mer RNA will be highly immune stimulatory by containing one of those motifs. It's very difficult to get rid of these by bioinformatics. What's a lot easier is to modify your RNAi to avoid stimulating these TLRs. This is showing the sequence of a single immune stimulatory RNA with a single stimulatory motif in it, just the UUGU. So that induces immune stimulation. But just making a single 2'-O-methyl modification inside that motif really eradicates the stimulatory effects over this dose range. And even if you go outside the motif, so here's the motif, if you make a modification at the very first base, far away from it, that does not eliminate the immune stimulation. But if you make a modification closer to the motif, it does eliminate the stimulation. So the good news is it's not that hard to get rid of the immune stimulatory effects of RNA, at least reduce them. But you do need to be aware of it, and you need to do the right experiments for it. So the issues that control this are sequence, the chemical modifications that you use. I didn't really get into all of the secondary structures. The other thing that's important is what delivery system you're using. Different delivery systems can actually avoid the immune stimulatory effect. And I don't want to be advertising for any particular systems, but it's probably good to screen these. Probably what's happening is that some delivery systems deliver their cargo into the endosomal compartment where the TLRs reside, and others either are delivering into different endosomal compartments or perhaps bypassing them, although that's been extremely controversial. Every time someone claims they have a delivery system that bypasses the endosomes, it turns out to be wrong. So I don't know, but empirically, you can certainly see big differences there. Now, the issues in evaluating RNA, there is so much junk in the literature. There are so many claims in the literature that particular RNA effects are not inducing interferons or other things, and they're simply wrong. And what tends to happen is that people don't use the right controls, they don't use the right time points. You see again and again in the literature, people use a particular control RNA that's sold commercially. And the reason everyone uses that is because it doesn't contain any of these immune stimulatory motifs, and so it gives you nice, clean results in your assay and makes you think, oh, wow, I'm such a good scientist. But really, you need to be aware of these. You need to use appropriate controls, because otherwise you're going to draw false conclusions. Mice do not have a functional TLR8, and so screening in human cells is an easy way to get around that. But what often happens in mouse experiments is that people will inject mice with their RNA, and then they draw blood the next day at 24 hours. Well, the spike in interferon and cytokines in mice is about a 4 to 6 hour peak. By 24 hours, it's back down to baseline. So, obviously that's pretty useless. And then using the wrong readout. People will sometimes transfect their RNAs into cells that don't express TLRs and say there's no interferon effect. This isn't rocket science, but it happens again and again. So, sequence selection, turning now to that. Ideally, you want to have a bioinformatic tool that analyzes the genome of humans and the species that you want to use for your tox studies, and then tells you what sequences are conserved between them all and which ones are going to be the right ones to go after. It's, of course, really not quite that simple, and you also want to exclude these other effects. My understanding is that the algorithms for predicting optimal RNAi sequences are really pretty good. I have not seen any published tool that has been independently validated for antisense sequences to predict the optimal sequences. You can certainly predict sequences that are likely to work with antisense. With the right chemistries, most sequences will. But what most people do when they're leading a drug development program for oligos is they just make a whole bunch of oligos and they screen them to find the best ones. I don't know of any better way of doing that. So the chemistries, Mano talked about these before. Chemistries can make a huge difference in your success and in what you see. They impact the efficacy, the uptake, and the safety. And I just want to show you some data from a study that we did back in my Pfizer group. Rob Stanton was leading this effort, where we looked at a number of different targets. This is glucocorticoid receptor. And what we did was we picked a couple of different sequences, 3 different sequences against glucocorticoid receptor. And then for each of these, we made a panel of oligos where we started out with a 14-mer and we made a 383 design. So LNAs are in the yellow here at both ends. And then we reduced the number of LNAs, or we substituted in 2'-O-methyls or 2'-fluoros or methoxyethyl, or we tried shortening the oligo by 1 base at each end. And then we looked at the percent knockdown, and we looked at a whole bunch of other things too. But basically, if you have a good 383 gapmer, those are active at the 2 concentrations here. But as you change the chemistry, you get all kinds of other changes in the activity that are really not predictable. And the changes in toxicity were striking as well. In general, having more LNAs in an oligo seems to be correlated with more toxicity, but it was very unpredictable, and making just a single change at one position could make huge changes in the activity of the oligo as well as in the safety profile. We were not able to come up with any bioinformatic way to predict that, but hopefully at some point there will be one. In the meantime, I think people are just experimenting and trying different things. So this toxicity studies, obviously the goals of these are to characterize what you're going to see. The difference, one of the differences from small molecules is that because you're targeting a gene and the gene sequences differ between different species, you may have to come up with a surrogate sequence to have the ability of an oligonucleotide to knock down the target in your mouse. If your human antisense candidate doesn't work in mice, then you need a mouse surrogate, or you may be asked to provide a mouse surrogate. And this will allow you to address the exaggerated pharmacology. Now, on the other hand, the surrogate is not going to predict the off-target effects of your human oligo. So basically, you could wind up in the situation where you have to do essentially 2 full tox studies, one on the human oligo to try to characterize its potential off-target effects, but of course, those might not be conserved in the mouse either, and the surrogate compound that you're having. So this is the approach that many companies take. They try to avoid it if possible, naturally. There are some obvious drawbacks to that. The surrogate might not be as good as your human candidate. It might be better than your human candidate at knocking down the target. And so how do you handle all of those? This is a discussion with the regulatory authorities, and I think you just try to let the science guide you there. So the approach then that's often taken in safety studies is to do screening studies fairly early, in vivo screening studies, to try to weed out the bad actors. So you may do an in vitro screen where you screen hundreds or thousands of sequences. You try to get them down to maybe a dozen, maybe dozens, and then you just screen those in rodents. And for example, some people dose mice at 100 mg per kg or 200 mg per kg for just a fairly short time, maybe 2 weeks or something. And then anything that shows up as toxic in that, you just kill, and you go on only with the sequences that are not toxic in that kind of a screen. You want to do some kind of pre-screen before you go into your GLP safety studies, because those are the expensive ones, those are more time-consuming. Ideally, before you go into the GLP studies, you already know in rodents, and maybe even in primates, that your sequence is going to be safe. And your goal in the GLP studies is really to establish the dose-response relationship of the toxicity. There is going to be another talk in session 5. That whole session is on safety, and I think it should certainly be an interesting session for everyone. The responses of the species in safety studies they tend to differ from humans, especially in rodents. Rodents are well known to over-predict inflammatory reactions from humans. And so the findings from rodent safety studies, the regulatory authorities are all aware of that, they're used to seeing it, and you just, you don't want to overreact there. So let me turn now briefly to ADME, and really there aren't that many things that are unique about oligos in ADME. Analytical methods are slightly different from what you use with small molecules. But for a particular chemistry, the ADME tends to be about the same. Now it may be length dependent. Shorter oligonucleotides will have less protein binding than a longer oligonucleotide in general, and so that can change the relative distribution between the kidney and the liver. But otherwise, it's not that much of a deal. Usually, you don't need that much ADME data early on. And in general, the charged backbone oligos are degraded fairly predictably by base clipping. LNAs and other things can change that a bit. Elimination is fairly straightforward. Uncharged backbones, interestingly enough, do not seem to be degraded, and so it is going to be interesting to see what will happen with those. So let me turn now to PKPD, also pretty quickly. And the goal, of course, with PK is to understand how exposure and the pharmacology of the oligo relate. And in this case, there really are some differences from a small molecule. With small molecule, you focus really on the plasma AUC, and you expect that that's going to be predictive of what's happening in the other tissues, and those relationships are really worked out pretty well. With oligonucleotides, that is not the case. Oligos typically are cleared from the plasma fairly quickly, within minutes to hours, and their lifetime in the tissues is much, much longer. In the liver, certainly weeks to months. In other tissues, months or even longer. Brett showed the data, for example, on the CNS, where effects are extraordinarily long after a single exposure. So the tissue concentration is really what you care about with oligonucleotides, but the relationship to the plasma concentration is only very indirect. Now, it can still be a surrogate for tissue concentration, and in some cases, people have been able to show that, but there is a difference there. These are some data that were provided very kindly by Akshay Vaishna from Alnylam, just showing the PK for one of their siRNAs that's in the clinic, and making the point that there's a very nice dose response from 2.5 mg per kg, where you get about a 60% knockdown at those doses. And then as you go up to higher doses, there's the expected dose proportional increase in activity. I guess you can't really say it's dose proportional from this, but the activity goes up as you increase the dose, not surprisingly. And another thing that's nice is that the pharmacology, or the PK in primates and in humans, is pretty much the same, so you can use the primate data to predict PK in humans. It would obviously be much more challenging if that were not the case. So let me turn now to clinical development to finish up, and really can only just hit a few high points here. The kinds of studies that are required for oligo clinical trials are really the same as the studies that are required for most other trials. Genotoxicity is normally done. It's always negative, and I'm not sure why. It's really more of a box checking thing than anything else. Same thing with safety pharmacology. It's just very uncommon to see anything in the safety pharmacology studies with an oligo. They don't tend to cause prolongation of QT or anything like that, but we still need to do those studies. And then, of course, characterizing the toxicity in a rodent and a non-rodent species usually, perhaps with a surrogate compound, getting at least a little bit of pharmacokinetic data. The CMC definitely is an important part of any package because you're usually dealing with a chemistry that's already pretty well established. These techniques, and you're probably getting your oligos from a contract manufacturer that has that well in hand. Usually that's not too challenging. And then, of course, putting together a clinical protocol. Now, the toxicities that you expect in clinical development are largely inflammatory, immune in nature. And so I did want to come back and talk about that a little bit more. This is another slide from Art Levin. The PTT prolongation has been seen at relatively high doses, especially just with phosphorothioate oligos. There's been some very nice work done figuring out the mechanism of that prolongation. This has not been clinically significant, but anytime you see that, it is something that regulatory authorities may ask about. And so that now is pretty well understood. That has not really been a serious toxicity in humans. Complement activation occurs with phosphorothioate oligos at very high concentrations in monkeys, and it can be dose limiting in monkeys if you give the oligo very rapidly by IV push. It really has not been an issue in humans, except with lipid nanoparticles. Lipid nanoparticles in humans, you very often see infusion reactions, complement elevation, and you may not see any of the cytokine activation in humans who are having significant infusion reactions. So complement-mediated effects can be a very important cause of the toxicity in human clinical trials, especially with these lipid particles. And anaphylactoid responses can be a part of that too. I think these are probably related. In humans with lipid nanoparticles, you tend to see either very early infusion reactions that occur while the patient is still in the clinic or wherever they're getting the infusion. They can develop rigors, chills, they can even drop their blood pressures. And then there's a second set of responses that patients can have after they've gone home, which can be hours later or sometimes even the next day. Complement may be involved with both of those. It's really not fully understood. And a lot of work has been going into analyzing exactly the mechanisms there. They do appear to be, immune mediated, and they may or may not be sequence mediated. Certainly, whatever lipids are involved in the particles, those may make a difference as well. Injection site reactions, all subcutaneously injected oligos have been related with injection site reactions. And with many clinical programs, that has actually been a limiting toxicity for patients. Patients have dropped out of clinical studies in large numbers because of those injection site reactions. I say large numbers, in some cases, 10, 30% of patients stopping treatment with their oligo because of the injection site reactions. Now, these are, of course, dose and concentration dependent. So although that's been an issue in clinical development up to this time and with the early chemistries, as we improve the chemistries and develop more potent compounds, the hope is that we can reduce doses and injection site reactions will be less of an issue. The other point I want to make with injection site reactions is the duration of therapy. For clinical indications where a patient only has to get a few doses, it may not be an issue to have injection site reactions. If you're doing weekly dosing for 3, 5, 10 years, we're more in uncharted territory about what will happen with those injection site reactions. Will there be chronic changes that will develop that will be even more worrisome than what we've seen so far? We just don't know. But I definitely want to highlight that. Reductions in platelets, slight reductions, no big deal. Those can be just redistribution, a splenic enlargement. But there's also been occasional cases of high-grade thrombocytopenia, very significant reduction in platelets. And there's at least some evidence now out that those are mediated by antiplatelet antibodies. I think there's more work to be done there. Fortunately, that is an uncommon, very uncommon effect compared to injection site reactions. Now, kidney changes in humans, these are in rodents, these have been studied very well, and primates. We do know a fair amount about what's happening in the kidneys and glomerular lesions. We don't know as much, obviously, about what's happening in the kidneys in humans. In most oligonucleotides in clinical development, renal effects have not been a major issue. LFT elevations have occurred more commonly. But for certain clinical indications where you have to push to high doses with the chemistry, then there is more concern about those higher doses leading to more renal accumulation and potentially to long-term changes. Vascular changes and actually vasculitis, this has been more in animal safety studies at very high doses. I'm not aware that this has been an issue in human clinical development so far, but it has occurred at least in the rodents. So finally, I'm just going to go on to my conclusion slides here. And, well, I guess it is worth making a point, again, just reiterating to be very suspicious about these immune and aptamer effects in your clinical development. Oligonucleotides have a very wide range of effects, some of which are sequence dependent, some are sequence independent. The off-target effects are less with the steric blocking oligos. Watch out for these certain motifs, but also in the case of RNA, you can reduce these in many cases, and certainly in preclinical development and also in clinical development, I think you want to have a fairly high index of suspicion for the immune activation, and there are certain assays that you can perform for those. For the preclinical development, the assays that we like to do are in vitro human PBMC assays, or some people use a whole blood assay. I'm not sure one is any better than the other. You just culture the cells for 1 or 2 days, you take the supernatants, and you assay them for the cytokines and chemokines that are known to be induced through these immune pathways, and I've listed some of those there. In vivo in mice, you draw blood at 4 to 6 hours, perform the assays. And then in vivo in humans, the time points are delayed slightly. Humans have a peak cytokine and chemokine response at about 24 hours, and those again are listed here. And that's my last slide. Thanks very much.